I'd like to start, of course, by acknowledging the traditional owners of the land on which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects to the elders past and present, any who may be here today. It's my great pleasure to welcome Associate Professor Chris Tonkin to give the seminar today. When Chris asked me yesterday to introduce him today, I was, of course, very humbled and excited to do so. I suspect you'll regret that decision in about 3 minutes, Chris. Chris has a very decorated scientific history for someone so young. He has, first off, started his PhD with Jeff McFadden at the University of Melbourne, where he studied the mechanisms of how malaria parasites traffic proteins into an essential organelle called the plastid. And at that time published some really nice work, including first authorships in Science and PNAS. And then Chris was awarded an NHMRC fellowship where he joined Alan Cowman's laboratory here at WEHI. And he worked on antigenic variation and the epigenetic control of that, which is a critical feature of how malaria causes disease. And yet again, Chris published some really important papers, including in PLOS biology, and also contributed to other work from the Cowman lab. It was around this time that I got to know Chris. Chris has been here for 16 years, and I've known him for 14 of those years. I think really at that time in the Cowman lab, Chris was strategically thinking, as he usually does, about what he'd like to work on. And he identified a parasite called Toxoplasma gondii, which you can see beautifully here in this opening slide, as a medically important human pathogen in its own right, but also a parasite that's really useful for studying as a model the biology of infection by apicomplexan parasites, which include the malaria parasite. Chris studied under the tutelage of David Roos in order to learn how to culture Toxoplasma and perform really elegant conditional genetics, which he's brought here to Australia and used really successfully since establishing his own lab here at WEHI in 2010. So Chris has been really successful in his time as a lab head and really somebody I've looked up to, and I'm very grateful for that opportunity. I know most people in this room know Chris, but for those of you who don't, I can assure you you're in for an absolute treat today. So welcome up to the stage, Chris, for your 4th Wednesday seminar.
I don't regret that at all, Justin. I'm going to get you to introduce me more. Okay, thanks everyone for coming to hear a talk on parasites. So I'm going to tell you about this beast here. Well, actually, it's multiple beasts. This is Toxoplasma. It is a banana-shaped parasite, and it's blue. In here is its nucleus, and its circumference. It's got a red stain here. And a protein that I'm going to tell you about today is the green stain here. So these parasites are replicating within a host cell. Okay, so to sort of provide a bigger context, I just wanted to step out from what you guys might already know, which is about malaria parasites or Plasmodium species. So Plasmodium belongs to the apicomplexan, the phylum Apicomplexan, which actually comprises of some 3,000 species, and many of which are obligate intracellular parasites. But also in this same genera is a series of medically important species. One of them is called Cryptosporidium, which causes severe diarrhea. And I'll tell you a little bit more about this right at the very end of the talk. There's a group of agricultural pathogens here, Eimeria, that causes losses in chickens, called coccidiosis. Babesiosis and Theileriosis are both cattle parasites. This one, quite interesting, Theileria, when it invades the leukocytes here, it transforms the leukocytes and gives the cattle a cancer. And when you cure those cattle of Theileria, the cancer regresses. And of course, my favourite here, which is Toxoplasma gondii. So first, I thought I'd just give you a basic introduction as to what toxo is, and then we'll get on to the data. So toxoplasmosis, so about 30% of people are infected with toxo, and it can be as high as 80% in some nations. And what happens, the reason we're all infected with that is we often are exposed to the parasite, and this is usually from ingestion of raw or undercooked meat or vegetables that are contaminated with the cyst form, which is secreted by the cat. So if you've been pregnant before and you've been told by your doctor to stay away from cats, this is why. This is the parasite why. So here is the bradyzoite form, right? This is the latent form that lives in the muscle and the central nervous system of its host, including us and other animals. And then what happens is that when we ingest this, we get this differentiation process into the tachyzoite here, okay? So you can see, just to orient everyone here, this is infecting a fibroblast here. They're about 7 microns long, and we've got 1, 2, 3, 4, 5, 6, 7, 8 parasites, right? So they go from 1, 2, 4, 8, 16, so on. Okay, so you get this differentiation process, but then if you've got a healthy immune system, so after you get tissue dissemination and destruction, you get mild flu-like symptoms often, and with the immune pressure that we have, we then get this differentiation back into the bradyzoite form, which then lives for our life. So our body cannot clear bradyzoite forms, and no one understands why this is. And of course, for most of us, that's not necessarily a problem. Okay, and these forms are, as I said, in the cysts in the muscle tissue, and stay for us for life. But of course, when we get immunosuppressed or our immune system isn't functioning properly, people that are undergoing chemotherapy, for example, can have these reactivation and get these toxoplasmic lesions in their brain, which are nasty. And I'll show you this in a second. So here, as I said, so what happens often is that if you're infected first when you're a mother, the toxo can transverse the placenta, and you can get these nasty congenital birth defects here. In this case, it's hydrocephaly. But what has really been unappreciated until really recently is that toxo in some nations, especially Brazil for some reason, causes this enormous amount of progressive blindness. And so you can see here, this is the retina, and we've got this plaque forming. So you get the parasites lodging in the retinal tissue, and then slowly over time, even though you do have a healthy immune system, you get this slow destruction of the retinal tissue and therefore blindness. And in especially in the age of the AIDS epidemic, we also got these individuals here, which were getting these large lesions in the brain. But what is also curious and not really understood also is that Toxoplasma can change the behaviour of infected mice, and this is thought to do with, it's got to do something with the parasite lodging in the brain, and also has these interesting correlations with mental illness. And this is some of the work that Shiraz has been doing in the lab, and he'll probably tell you a little bit more about that another time. So this is the apicomplexan parasite. They all are very, very similar. You could put here, this could be the malaria parasite, this is, could be Toxoplasma, this could be Cryptosporidium, it could be any of them. And this is what they look like. Okay, so the only difference they have is that on their surface, right, they secrete proteins that are specific for the host cell they want to invade, right? So of course they're a eukaryote, so they have a nucleus, and they have an endoplasmic reticulum and a Golgi, and a mitochondrion. And they also has, as Justin said, what I would do my PhD on eons ago, was that it has this relict plastid that's non-photosynthetic anymore, which seems to be very important for the biology of these pathogens, and actually shows that this group of pathogens evolved from free-living alga. But then there's these other components which are apicomplexan specific, and this is where they derive their genera from. And this is this membrane complex that sits underneath the plasma membrane here, called the inner membrane complex. And then it has these other apical organelles. One here is called the micronemes. I'll talk about these a bit more later, which harbour the adhesins. And then they get exocytosed onto the surface of the parasite. And then we have these rhopteri, these large club-shaped organelles, which are involved in the formation of the vacuole as the parasite penetrates its way through. And then we also have the dense granules, and they harbour effectors generally, proteins that are secreted from the parasite, which then cross the vacuolar space and affect the host cell in which they survive in. Okay, so here is the complicated life cycle of toxo, going between mammals and felines and in the environment. And I don't really want you to pay attention to the details, but I wanted you to just to illustrate where my interests have lied over the last 5 years. And that is what I find really interesting and curious, is that these pathogens have to sense where they are, whether they're intracellular or extracellular. They have to sense what host they're in. And then when they get there, they have our immune systems to deal with. And they wouldn't be pathogenic if they couldn't overcome those immune defences. And every time I sit in these Wednesday seminars and I hear people talk about, innate immunity or even adaptive immunity, I'm amazed to understand that actually these pathogens, anything that's a pathogen, by its very nature, has to overcome all these different defense mechanisms. And I find that really fascinating. But nevertheless, what I'm going to tell you more about today is understanding the sensing mechanism. How does the parasite know where it is? How does it know when to differentiate into bradyszoites? How does it know when it's intracellular to start replicating, or when it's extracellular, it's time to go and find a new host? Okay, and this can be the same said as it's changing its life cycle stage as it goes through all these different sexual stages here. How does it know when to go to differentiate into sexual forms? How does it know when to package itself, so on and so forth? But what I'm going to do for the purposes of our research is I'm going to talk more about just the lytic stage, because that's the stage that's most amenable to our understanding, okay? So this here is the acute stage, the tachyzoite, and I've shown here one host cell bursting, its vacuole and the parasite's bursting out of the host cell and has to migrate and find a way of getting inside the next host cell so it can then settle down and start replication again. And so this is what we've been really interested in. And so just to orient you, this is a movie that I've got of this whole process, right? So here we've got fibroblasts infected, and there's 1, 2, 3 vacuoles full of parasites. There's probably 16 to 32 parasites in each. And we can give them a little tickle, and then which allow them to activate their motility. So look what happens when this occurs. Okay, so you get parasites egressing out of the host cell, and they all move away, and they try and find a new host cell to invade. Okay? So what we've done over the last 5 years is we've worked on several different aspects of this, and I just wanted to provide a snapshot first of what we've done, and then I'll home in on one particular story. So one of the really important things for this motility process is this motor. It's a myosin-based motor, and it sits between these inner membrane complex and the plasma membrane here. It attaches the host cell receptors basically to an actomyosin motor system. And we discovered a little while ago, we actually discovered what the full myosin was, okay? And so before then, no one ever knew what it was. And so it's only now, because of these discoveries, is it's found that actually that people have been able to understand the biochemistry and the structural biology of this particular motor. And we've also worked with Ethan on adhesins, how some of these adhesins are glycosylated and their role in motility invasion. And a serendipitous finding was also the regulation of metabolism. What we didn't fully appreciate is when these parasites are changing from intracellular to extracellular, they need to alter their metabolism. They need to have stores of energy in order to move, to power that motility. And we found actually that one of the pathways that I'll tell you a little bit about today is controls a myelopectin or starch synthesis. And if you knock this gene out, you get this enormous accumulation of starch. And you can see these parasites here are defunct, right? They're just full of starch. And this actually was really interesting because it's led us to patent some of this work as an attenuated strain for vaccination purposes, because these parasites just cannot persist. Okay, so we've also been working a little bit on host cell manipulation. I touched upon this before. This was headed by Michael Coffey, as many of you know, and also has been work done by Simona Sazova. And she'll give a talk about her work on the bradyszoite stages of this protein export. But basically, you can see here, this is an infected host cell down here. Red demarcates the parasites. Blue is the host cell nucleus. And as you can see, we've got this protein that is being exported from the parasites and imported into the host cell nucleus. And this is really critical for pathogenesis. In fact, we found the way that these proteins were trafficked, and we found proteins that were specifically involved in the trafficking of these proteins. And so we knocked them out, and they rendered the parasites completely avirulent and unable to modify their host cell. And so what I'm going to do today, though, is I'm going to talk to you about these two separate but interlinked programs that we've had running, okay? So first of all is discovering how parasites activate motility, right? So as I've said before, they need to know when it's time to leave the host cell. They must receive signals in order to do that. And then they move and then they invade, but then they also need to quench that motility. They need to stop and then know when to start replicating, right? And understanding this process has been, of course, it's applicable not only for Toxoplasma, but understanding how generally these apicomplexan parasites sense their environment. So here I'm just going to show you this conserved process of gliding motility in this group of parasites. So here we've got the Plasmodium species in the liver stage, which is the stage that Justin works on, And they are moving. They need to, after they've been injected in by a mosquito, they need to find the liver. And they obviously need to sense their environment to do so, and they have to activate motility in order to do so. Okay? You can also see here Cryptosporidium, this gut pathogen, which I'll tell you a bit more later. It also has gliding motility as it glides across the glass slide here. I've told you a little bit now about Toxoplasma. It's here is invading a fibroblast, you can see here. And also the work from our division has also highlighted really important mechanisms as to the merozoite, which is a tiny little parasite here, as it buries its way into this host erythrocyte here. They all require motility, and they need to know when to activate it. And I've shown you this Toxoplasma egress as its host cell, so I won't do that again. So the question is, why do we care? Why do we care about understanding how these parasites sense the environment? And that is because, this audience well knows, that signaling pathways are really eminently druggable, okay? And so I've highlighted here actually several bits of work from members of the audience here to explain this and to draw your attention to this process. So here we've got intrinsic and extrinsic apoptosis. We've got innate signaling through the inflammasome here, and we've also got the JAK-STAT system here as well. And people use this, and as we all know, that this is really eminently druggable. So what about apicomplexans? Can we use this information to find new drug targets, to find new therapeutic interventions? And so here I've just put here a big black boxes because that's largely where we stand at the moment. Although some kinase inhibitors that are part of these signaling pathways in some way which we don't understand are really under development. And so we need to understand how these signaling pathways work to fit into context how those drugs work and how resistance might be curtailed. Okay, so coming back to Toxoplasma and what we know already about how these parasites sense their environment. Okay, so this is an infected host cell here. We've got Toxoplasma here, and there's two ways that they sense their host cell, which I'm going to tell you about today. One is, it seems that they sense the pH of the vacuole they're living within. And we think that that's because as the immune system attacks and lysosomes start being deposited on the vacuolar membrane, you get acidification of the vacuolar space, and the parasite needs to know when to exit after immune attack. The other curious one is changes in potassium levels, potassium concentration. And so I envisage this might occur when you've got a cytotoxic T cell and you've got and they're attacking a host cell that's infected, and then you've got changes in the membrane potential. And we know that NLRP3 senses the changes in potassium level, and this is also probably a trigger that the parasite wants to get out of that dying host cell. So changes in potassium concentration and changes in pH are what these signals are. Okay. But also wanted just to quickly mention that these signals, what I'm interested in first is what are those signals, how are those signals received, and then how do those signals transduce to activate these processes that I'm telling you about. So I've told you here about toxo, but I also wanted to bring your attention here is that this is the Plasmodium falciparum, and when it is taken up by a mosquito, you get a drop in temperature and a change in the metabolic environment. It needs to know when to now change into the sexual forms. And as soon as it receives those signals, it changes into these male and female forms here. So this process is equally applicable to these stages too. So what needs to happen after these parasites receive the right signals? So first, you get secretion of these adhesins onto the surface, which I was telling you about here, and they get secreted so they can bind tightly to their host cell. And then they also have to activate this actomyosin motor here, because the adhesins come along and they attach to this motor here, and the adhesins get dragged backwards through. And so what we do know, and what we knew at the time when I started my lab, is that one key feature stands out that we knew, and that is that calcium signaling, cytosolic calcium signaling, seems to be really important for this process. And we knew that because if you use an ionophore, which some of you might be wary of, is that it makes calcium permeable to membranes, and when this happens, when we add this ionophore, this is exactly what happens to intracellular parasites. They receive that signal, and then they egress. So it says that calcium signaling is really important for that process of activation. So the first thing we needed to do was try and develop tools to understand and to measure calcium. And the way we went about that is that we developed this calcium-based biosensor called GCAMP. Okay, GCAMP is a circularly permuted GFP, and it has calmodulin attached to it. And in its native state, it's non-fluorescent. Only when calcium is present will the conformation change and you get fluorescence. Okay, so look, this is what happens here. When we add the ionophore, you get increase in fluorescence. The parasites have high calcium levels in the cytosol, and then they activate motility. And the good thing also is that what we can do is that we can use a fax-based method too here to monitor these changes in fluorescence. And we can see here, as we titrate down the concentration of the ionophore, we get less and less signal, okay? So it's quantifiable. This is a kinetic graph on fax here. So we have the parasites, we put the parasites under and they have resting levels, and then we inject in one of these agonists and you get this huge rise in level of calcium over time here, as you can see. Okay, so how does environmental sensing link in with calcium signalling? This is the first experiment we needed to do, and here I can show you just with some quantitative imaging done in collaboration with Kelly Rogers, is that if you have high concentrations of extracellular potassium, that is mimicking what they normally see in their host cell, this is what you see. Okay, you see very few parasites that have high levels of GFP. Most have low, i.e. blue levels of GFP, and they may remain quiescent. But as soon as you change that buffer and you drop the level of potassium, it thinks that there's something wrong with the host cell, and this is what happens, okay? You get high levels of high levels of calcium occurring, and the parasites are moving along the glass slide. Okay, so as it senses the environment, it's transducing through calcium signaling, okay, to activate motility. Okay, so this is our model. So what are the signals? So I've told you here that the signals can be changes in potassium and changes in proton concentration, or pH. And then and I've told you here also that it transduces through this cytosolic calcium signaling, and that's what's required for activation of motility. Okay, but what I also wanted to draw attention to you back is also showing that this is also applicable to Plasmodium species and probably all the other apicomplexan too, because we know that these changes that are seen by Plasmodium, if they're taken up by the mosquito, are also transducing through calcium signaling as in order to activate that next stage of differentiation. So the signaling processes are largely are not necessarily conserved, but they have very similar commonalities to them. Okay, so what I'm going to do for the remainder of the talk is tell you about two stories in which we have developed over the last 5 years to understand this point here. How are the signals received? Now, this is really important because these parasites don't have GPCRs and they don't have receptor tyrosine kinases. So they do things differently from every other mammalian system. Okay? So I'm going to tell you a story about one about cyclic GMP signaling and the other one about cyclic AMP signaling. Okay, and how these signals are integrated, how they relate to calcium signaling, and how they're involved in sensing their environment. So if anyone that knows, and I don't think anyone works on cyclic nucleotide signaling in the audience, but this is how it occurs in mammalian systems. So you have these receptor-based tyrosine kinases that sense things like peptides and nitric oxide in mammalian cells, and that transduces there's signals received, and then you have this guanylate cyclase which makes cyclic GMP out of GTP down the bottom here. So you get this local rise in cyclic GMP concentration. Cyclic AMP works in lots of different ways, but here's one of them. You have a G protein coupled receptor which is receiving a signal. It activates an adenylate cyclase here which makes cyclic AMP, and then that is all linked up with a kinase here, this protein kinase A, which binds to those cyclic AMP molecules and activates phosphorylation. Okay, so then we thought, well, actually here I'll explain it a little bit more first. So here we've got this guanylate cyclase here. It's making cyclic GMP from GTP, and as it gets activated, you get cyclic GMP concentration rising in the parasite, and then you get this activation of this protein kinase G that then phosphorylates downstream targets. But then when that signal is finished, when you don't need that signal anymore, then you have a diesterase which then breaks down that cyclic GMP. So this dynamic process of synthesis and degradation, and the fine balance between those two is what dictates the level of cyclic GMP. So one of the first things that we... oops, there we go. I just illustrated that. And then one thing that we did do, the first thing we did is we worked with some chemists and we found a very potent apicomplexin-specific inhibitor of this PDE. We call this BIPO because it's got a long chemical name I can't say. This is its structure here, and we published this because it showed to be a very important tool for the understanding of these signalling pathways, which is now used by a lot of labs. Okay, so also what I wanted to mention too here is we have this other tool compound, or called compound 1, which has been developed as a specific inhibitor for this protein kinase G. And interestingly, this compound is highly potent against a range of apicomplexin parasites, including malaria. So this is what happens. So this is our first study. We're looking at what BIPO does if we activate. So you can imagine you're blocking the phosphodiesterase, you're allowing for the accumulation of cyclic GMP. What happens? How important is cyclic GMP for parasite motility? So here, I've shown you again, this is going to be another movie where we've added this BIPO compound to infected host cells and to see what happened. Does it activate motility? Does it somehow suppress it? What happens? And you can see also if we block the breakdown of cyclic GMP, therefore allowing for its accumulation, we also get activation of motility. And we also know that this is important because if you add compound 1 here, which is blocking PKG, and you add BIPO to allow for the accumulation of cyclic GMP, the parasites don't egress. All right? So that's telling us that the cyclic GMP pathway is really important for the activation of motility in Toxoplasma too. And this also turns out to be true for malaria parasites. So how does it relate to calcium signalling? So we thought maybe this is maybe it's acting together in concert, maybe they're separate. What happens to calcium signalling if we activate this cyclic GMP levels using BIPO? And again, we used our GCAMP expressing parasites here. You can see here their resting levels. They've got blue or low levels of fluorescence. And then when we spiked in BIPO, this is what we saw. Again, we see this rise in calcium signalling. And this is also very specific because if we add the compound 1, we don't get this effect occurring. We don't get a rise in calcium levels, okay? So what that tells us is that they're linked and that cyclic GMP signaling is probably acting upstream of calcium signaling. The cyclic GMP signaling is somehow activating and allowing for release of calcium from calcium stores in the parasite, and that then allows for activation. So then when we looked further upstream, what's happening? How are these parasites sensing their environment? Okay, so as I said before here, they are often, in mammalian systems, they have these membrane-bound receptor tyrosine kinases. These parasites don't have it, but instead they have this enormous 477 kilodalton protein here, which has these guanylate cyclase-like domains here in red. But instead, this is this transmembrane domains throughout the protein, but these transporting ATPase-like proteins here. And we thought, well, if they're sensing ions in the environment, maybe this is the mechanism in which they are able to activate the production of cyclic GMP. So we wanted to investigate this, and so what we did first is we tagged this protein using HA. And you can see here's a single parasite in this particular case. It's facing downwards. GAP45 is a membrane marker here in red. And you can also see this, so the HA signal is really at the very tip of the parasite where we'd expect that a lot of the sensing has to occur. So what we can see, so that was very interesting. And so what we can do in Toxo is we can make conditional mutants quite easily, and we did this by doing a promoter swap and basically making a TET off conditional knockout for this Toxoplasma gondii guanylate cyclase. And we can show here that what we do, just to validate this system, we can show, interestingly, we have this guanylate cyclase seems to be in multiple forms. We think this must be proteolytic processing of this particularly large protein, although we don't know. But then as we turn it off using this anhydrotetracycline over 36 and 84 hours, we can see here that we're depleting levels of this guanylate cyclase. So satisfyingly, what we're able to show also is that as you remove this guanylate cyclase here, then we also reduce the amount of cyclic GMP present in these parasites also. And here is a measure of gross growth of Toxoplasma, and it's called a plaque assay, a bit similar to those people that do viral plaque assays. You have a monolayer of host cells which stain purple with crystal violet, and then anywhere where a parasite is and has cleared and lysed the host cells that it's living within, then it creates a little plaque that's devoid of those of host cells, and therefore ends up being a clearance. So you can see here that only when we turn off this guanylate cyclase, these parasites can't grow at all, right? So this is essential for their growth. Okay, so what's it important for? Is it important for activation and motility and invasion? And so we can tease this apart in Toxo, and we can show here that, that the parasites that lack the guanylate cyclase are not very able to attach to their host cells particularly well. This would suggest that the amount of adhesins that are being released onto the surface is defective. And also their invasion capacity is greatly reduced as you turn this gene off here. So the guanylate cyclase and cyclic GMP signaling is probably very important also. This protein is particularly important for invasion and attachment. So is it also important for this cyclic GMP-induced egress that I told you about, this BIPO-induced egress? So here we can add BIPO in and measure the amount of parasites that leave their host cells, and as you can see, also it's required for efficient egress from host cells when you turn off GC. So it looks like that this guanylate cyclase is really important for all the major steps that we can see. But interestingly, what we found is that when we directly stimulated calcium signaling using this ionophore, is that we got really efficient egress. And so the way that we envisage what's happening here is that the calcium ionophore is bypassing the requirement for the upstream cyclic GMP signaling and allowing for activation to occur, therefore suggesting that these things, that cyclic GMP is also acting upstream. Okay, so what we've also done here is we can measure motility. So here we've got both parasites that have the guanylate cyclase and parasites where we've depleted the guanylate cyclase, and then we give the host cells a mild detergent treatment which punches holes in them. And then we look for their ability to move away from that site of infection and disseminate through the tissue or through the monolayer of host cells. So you can see here, I've shown you this before, you get this, this movement of the tachyzoites as they move away from their point of infection, looking for a new host cell to invade. So what happens then? If we turn off the guanylate cyclase, they simply can't move away, okay? So the guanylate cyclase is really important for all those major transition events that are occurring as the parasite's transitioning from one host cell to the next. This can be quantitated, of course, again with help from Kelly's lab, who have been really instrumental in helping us quantitate all the data that we've got, whether they be fluorescent signals or movement of parasites and speed. As you can see here, that as we turn off this guanylate cyclase, the average speed and the maximum speed of these particular parasites is also negligible. Really, this really represents almost Brownian motion. So as I said before, microneem secretion is super important because it's releasing the adhesins onto the surface, and microneem secretion is required for all these processes which I've been talking about here, egress, invasion, and motility. So we can monitor that because we can take the supernatant of these parasites and we can tickle them with some of these agonists, and we can collect the supernatant and say how much of those adhesins is present in the supernatant. And so here you can see when we have, when the guanylate cyclase is present here, this here is a micronemal marker called perforin-like protein, which I'll talk about a little bit later, is that when you stimulate with BIPO or A23187, you get lots of secretion from those micronemes. But interestingly here, when you turn off guanylate cyclase and you stimulate here, you get really very little levels. So this guanylate cyclase is required for those secretion of those adhesins which are required for motility. So is it required also for calcium spikes? Here, so we use our GCAMP expressing parasites, and we use our FACS-based assay, and we spike in BIPO at 20 seconds and then watch the rise that occurs over time on a FACS. And you can see here the range of concentrations that we use of BIPO. We get these we get a nice rise in cyclic, this is just in wild-type parasites, in the level of calcium that you see. But what happens is that when we deplete the guanylate cyclase, we get no calcium signalling at any concentration that we see. So this is required also for activation of calcium signalling. And we can just quantitate that there. But interestingly also, the simple, if you bypass it using this A23187, you get this rapid rise in calcium signalling, whether you have the guanylate cyclase present or absent. So linking it back now to how these parasites are sensing their environment, then we wanted to understand, could it be that this guanylate cyclase is required for their ability to sense their environment? And so this is a bit more of a tricky experiment to do with FACS, because what it requires, you need to change the buffer very fast in order to then put the parasites under the FACS machine and read them very quickly. So what we've done here is, you can see here, when we have low levels of, if we have high levels of potassium and we drop the levels quickly by this process here, we get activation of calcium signalling, right? And here, when you don't have guanylate cyclase, then you get no response at all. So guanylate cyclase is required for their ability to sense the levels of potassium that are in their environment. And this is also true for the levels of pH, and you can see that again, much the same way, is that we can get these parasites to respond to the level of changes in pH in their normal condition, but as soon as you deplete the level of guanylate cyclase, then that no longer occurs. So this is the model so far, that we get activation of these external environmental signals, which then activate this guanylate cyclase to produce cyclic GMP, and then which then allows for protein kinase G to become activated, calcium to be released, and motility to occur. Oops. So one thing then dawned on us is that even though this activation process occurs, what is the thing that's turning it off? How do you get from an actively motile parasite into an invaded parasite that can then initiate replication? And so there must be some kind of negative signals here to prevent these things, to prevent the motility occurring. And so what I'm going to tell you a bit about today and now is a bit of a serendipitous finding that we found during this process. And so here we've got our GCAMP expressing parasites here, and they're invading a host cell that'll loop back around again in a second. They've got high levels of GFP, and as soon as they invade, they turn red. And that's because they're also expressing M. cherry, and they have no GCAMP left. So what they do is they quickly quench. As soon as they've invaded, they stop moving, and they quench their calcium signal, and that allows them to start the replication process. And so again, with help from Kelly, we then were able to quantitate the level of fluorescence and derive a ratio between these two to work out how fast quenching was occurring of this calcium signaling to allow for replication to occur. And as you can see, it's rapid. It occurs very fast. Within 30 seconds of invasion, you get a loss of this calcium signal. And so whether this is one parasite or multiple parasites, you can see that we've got very low we get very low levels and reproducible levels of the calcium quenching after invasions occurred. And so we were also studying at the time cyclic AMP signaling and this protein kinase A, which is required to receive the cyclic AMP signals. And we tagged it and showed that this protein was found at the apical, at the periphery of the parasite, and therefore was in the right location to have something to do with sensing, but we could never find anything other than it binding to its regulatory subunit. So we could never find it binding it to adenylate cyclase, this ACAP protein, GPCRs, or anything else like that that would resemble a structure like this. And it only found its regulatory subunit, which also is found in the same location. And interestingly, what happens is that normally these PKAs derive their membrane affinity by dimerizing together using this dimerization domain. And apicomplexa don't have that domain. But instead they had this motif here, which is a guanine and two cysteines here. And we actually showed, using a range of mutants, that this particular motif was critical for its association with the membrane. But if you then mutated these different glycine or cysteine motifs, which are known to be acylation motifs, then this drops off the membrane. So this signal is required for pKa to be membrane attached. I won't go through that. So then we made some conditional knockouts, and you can just see here the validation of this process. And you can see we were able to deplete the amount of pKa as time went on. No plaques. This protein is absolutely essential for growth also. But obviously the biggest question is how and what's its importance. And so when we made this mutant, Rebecca Stewart, who was a PhD student at the time, said, there's something really strange about this mutant. And she said, I'm going to do some live cell imaging on it, and I'm going to try and work out what's going wrong. And so for all those that are sleeping in the audience, this is the most important point, is that this is the phenotype that we saw, which was highly unusual. So this parasite here invades, and then it leaves again. Okay? Here, it invades and leaves again. It can't recognise that it's inside the host cell. It can't sense the environment to turn off motility. We spent a lot of time trying to work out, was it a defect in invasion, or was it a defect or were they activating egress too early? And this is the single experiment which clinched it for me. We used a fibroblast line that has membrane-bound TD tomato. And we allowed parasites to infect these fibroblasts, and we were looking for their ability to make a vacuole around themselves. So what I want you to pay attention to is the fluorescent channel here, and watch what happens when the parasite invades, the membrane that surrounds it. So it invades, and you can see it clearly makes a vacuolar membrane around it. But then, out it comes. And this parasite also, if you look carefully, then re-invades this neighbouring fibroblast and does the same thing. So they're restless. They can't turn off motility. And we can quantitate that again with help from Kelly to look at how long it took for those host cells to collapse. So of course the question then is, how does that relate back to the model that I've been showing you throughout? Does it regulate calcium signalling too? And is it negatively regulating it? And the answer seems to be yes. So what happens here is these are just measurements of cytosolic calcium using fluoride dye, and we can show as we increase the concentration of extracellular calcium, the parasites either a resting state, or as you put in more calcium into the media, they are unable to regulate the level of calcium that's cytosolic. They have too high level. So then again, what happens using our GCAMP parasites? I've shown you here again, I've shown you this before, the parasites invade and they quickly dampen down their calcium signaling and they turn red. So here is what happens when we remove that protein kinase A. Watch this parasite come from the top here, this one. It stays yellow. It can't turn off its calcium signaling. It turns on and off, it can't regulate it, and it destroys the host cell it comes out. So it's unable to regulate the level of cytosolic calcium. And we can quantitate all this here again, as I've shown you before. This is the calcium traces the GCAMP traces, and you can see they're just unable to control their levels. Unable to control their levels of calcium signaling, of cytosolic calcium. And we can do lots of different measurements to try and quantitate these levels, whether it be how long it takes for the time it takes for 35% of maximum fluorescence, what is the fluorescent level at 100 seconds, and so on and so forth. And in every measure, they're defective. So is it really egress? And the way we addressed that is we made another mutant. The perforin-like protein in toxo is required for egress. These parasites have a perforin-like protein that sits in their micronemes and they secrete it, which then damages the vacuolar membrane and the host cell membrane, allowing for them to egress. And this is the mutant that was first described many years ago now. And you can see here the parasites activate motility, they just can't break out, right? So they're just circling around in there, right? They can't get out. So what happens now if we've got our mutant, but then we also knock out PLP1, right? The parasites invade, they're having trouble regulating their calcium signaling, but they cannot exit out again because we've blocked them. right? So it says that whatever's occurring is it's dependent, the pKa is negatively regulating the perforin and the perforin function in some way. Okay, and I won't go through that because I haven't got time. Okay, and I'll skip that too. So this is the model. The model is that we receive signals, these parasites receive their signals from the extracellular environment, and it can be potassium pH, or it can be changes in their host as they transition, they transmit from host cells, and they have to sense those signals, they have to transduce those signals, and they then have to activate their processes. And we spent some of my first time here as a lab head, I was working on how those calcium was transduced into phosphorylation signals to activate this process. And this is what I've been focusing on over the last 5 years. How do they sense? How do they transduce those signals? And I've shown you now that there's this dynamic interplay between cyclic AMP and cyclic GMP signaling somehow. We don't fully understand that, but they're relaying these extracellular signals through these two arms, which is allowing for the regulation of motility. And to put it in a different context, also, you can see here that I've shown you this arm here before, but that pKa and its regulatory subunit, which is binding the cyclic AMP, must somehow negatively regulate this process. So what I wanted to touch on just briefly is, again, come back to the apicomplexer and talk a bit about what we want to do in the future. Okay. So here, what we're going to do is actually translate some of these findings that we've done in Toxo, using the genetic tractability of it, and now focus on this other parasite called Cryptosporidium, which I'll have 2 slides on. So Cryptosporidium is a leading cause of diarrhea, especially in Africa. And there was a leading study come out of the Lancet a little while ago showing that it's actually the second leading cause of diarrhea in children under 5. So rotavirus being the most common, Cryptosporidium followed by these 2 other bacterial gut pathogens here. And this was done in both Africa and Southeast Asia, and you can see the citation. So this is a massive problem. Mortality accounts for 10.5% of 8 million deaths in children under 5, as you compared to malaria, 7%, and HIV 2. So it's really there's an unmet need for working on these diarrheal pathogens. It can cause malnutrition and stunted growth, and it's also responsible for outbreaks in developed nations, some huge as well. And we have plenty of outbreaks of Cryptosporidium in Australia as well. And it's a really major problem in immunocompromised individuals that get this sort of chronic diarrhea because of Cryptosporidium. So as we have an ageing population, this may also become really important. But the clincher is there's no drugs. So there's one partially effective drug that doesn't work in immunocompromised individuals, and there is no vaccine. And the reason why all this is the case is that no one's been able to work on this pathogen. It's been intractable in terms of experiments. But this has somewhat changed recently, which is really exciting. And so now this pathogen can be genetically manipulated, and there's new tissue culture systems coming online. Here is the microvilli from the gut. Here is this little beast, Cryptosporidium. It doesn't fully invade the cell that it's living within. It's called intracellular but extracytoplasmic. It creates this, wraps the membrane of the host cell around itself and has this sort of conduit here which exchanges something that we have no idea what with the host cell. How is it surviving there, right? How does it prevent the death of the enterocyte that it's living within? How does it invade? Lots of questions here that are really open. And so what I plan to do over the next 5 years is to try and develop these systems at WeHi. And there's lots of different reasons for that. But one is that I think that this pathogen is very interesting in the case of dysbiosis of the gut flora. It provides a really interesting infection model to understand the gut because it's invading the gut and living within there. We've got so many resources here at WeHi to try and understand that could allow us to understand that interaction. What I'm really fascinated, of course, is how this parasite prevents the cell death of the host cell that it's living within, and also how it's invading. And also, we have this really interesting bit of work going on in in the ID2 division, which is using finding drug targets that are involved in drug targets that are targeting proteins that are involved in invasion, we think. And these are the aspartyl proteases. And so I'm going to do is look at what role these aspartyl proteases play in invasion, how important they are within the life cycle. And the second thing I'm really interested in looking at is there's no drugs, but can we use the ubiquitin system to try and enhance the drugability? And so we've got some work with David Commander and other people around the Institute trying to see if we can at least identify the ubiquitin machinery, because there's nothing known about ubiquitin biology in these parasites. So I think we can use this as to try and enhance drugability and use Toxo to try and understand the ubiquitin system, what it's required for other than just protein degradation. So then what I'm really also interested in, what are the proteins? So we've been working a bit about how Toxoplasma manipulates its host cell, but I want to know now how Cryptosporidium does the same thing. What proteins are exporting? What are they doing? How are they preventing cell death? How are they skewing the immune system to allow for them to survive in the gut? So I think that there's some really interesting new techniques that we can use to try and understand how the Cryptosporidium is manipulating the enterocytes. And I think, so what I'll do is I'll leave it there and just, of course, mention, I'm not the one who's doing these experiments. These is the group, both current and past. I've had really talented PhD students which have driven a lot of this work. In fact, most of the work I presented today was done with PhD students. I've had multiple collaborations, which I've tried to show you at the start, really working a lot with Andrew. We've developed new proteomic techniques, working with Ethan to try and understand the role of glycosylation, with some of the chemists and immunologists as well, which has been really fruitful. There's some external collaborations which I won't go into, but also a special shout out here to Kelly. As you can see, our work is very image intensive, and we not only require to take those images and have microscopes working at their utmost best all the time, but we can knock on their door and say, Lockie, please help us. We need to know how to analyse this data. How do we quantitate it, right? And so that has been a really fruitful collaboration. So with that, I'll leave it and take questions.
Does Cryptosporidium also have a Brady state, or is it always an acute infection, or are there carriers? Or?
Yeah, so there's as I said, not much is understood about the biology because you haven't been able to do anything, which makes it really exciting, right? But what it does is it has an obligatory sexual stage in us as well. So it goes through an asexual stage, then it makes the sexual stage, and then it packages itself up into oocysts, which then you defecate out, or it can then continue the cycle on again. So the bradyzoite is the oocyst, which is really environmentally stable, and that's why we have the water sanitary practices that we do. Yeah.
So is the phosphodiesterase not able to end the cyclic GMP? You showed that you get a difference in transference.
So what you would expect is that you can stop the signalling by activating the phosphodiesterase. Is that what you're saying?
Yes, because you were looking at what happens, by what mechanism do the tachyzoids stop migrating and stop.
Oh right, yes, okay, yes.
So phosphodiesterase is not involved in that?
Could be, and it seems that we have evidence to say that the pKa actually phosphorylates the phosphodiesterase, and so it might be an activation signal. But again, how that works, how that signal is received is unknown, the cyclic AMP.
So again on the, I guess the potassium that's leading to the tachyzoids stopping in the cells.
Yeah.
So if you interrupt that process, then they don't stay, right? So, when you're looking at how you use their egress, do you think you're just mimicking the PKG mutant, and then you're actually, instead of measuring egress in some of your experiments, you're actually just measuring the inability to stay.
Yes.
In the cells.
Yes.
No, so is it really the natural egress signal?
So, no, we don't know. But what the working model, I suspect, is that if you want to be a motile parasite, you have to have high levels of cyclic GMP, therefore calcium signaling, right? And it's sensing the environment to allow to regulate that. But then when you invade, then I suspect what happens is that the levels of cyclic AMP then rise, right? And then it's the off switch. You say, I've got to turn off now, and therefore that allows to override that signal. But you're right, there's these nuances that we don't understand, and it could be actually that PKA signaling is regulating the basal level of calcium and not being dynamic. But it's really hard to tease that apart.
It was really good talk. I was interested in the directionality. So when you put the calcium on top of parasites when they're on the glass slide, they seem to move about, but they remained interspersed with each other. They didn't cross over.
Yeah.
Have you tracked any movement to see whether they do stay equidistant?
That is complete. Yeah. You might have to ask the students. I don't know about that, but they could. But what also is important to mention is that that's a glass slide. It's in 2D, right? And they have these different forms of motility. When you flatten them onto 2D surface, they'll go in circles. They'll sit on their bum and twirl. They'll helical, move at helices. But as soon as you provide a 3D matrix, then they move as a corkscrew, right? But as for whether they sense each other, I think it's probably unlikely, but no, we have no data on that, and it probably does happen. Yeah.
We've got time for one more. Is there anyone at the back or in the middle? Everyone at the front? Seems to be really keen. Did anyone else at the back also? No? David.
This protein kinase A has been targeted by many small molecules. Do the small molecules mimic your egression?
Good question. So we have only tried one, which is H89, which is, I think, not very specific. It doesn't work against the toxo version at all. But yeah, an interesting idea that we haven't really looked much into.
Okay, great. Let's just thank Christian.